Controlling your breathing are two respiratory centers in your medulla oblongata, which is this purple space right here. It's the med-ob. And I have one of those respiratory centers shown there with an oval shape. The ventral respiratory center, or the VRG, sets the basic rhythm of respiration. Within the VRG, there are two collections of neurons. There are those that cause inspiration, known as I neurons, and there are those that cause expiration and are known as E neurons. Both sets of neurons contain pacemaker cells, and those are cells similar to those in the AV and SA nodes of the heart. Those were neurons that spontaneously depolarized, right? They refused to have a resting potential. And when they do depolarize, they inhibit the other collection of neurons. So the I neurons firing inhibits the E neurons from firing for about 2 seconds. Then the E neurons fire and they inhibit the I neurons for about 3 seconds. And this 2 second, 3 second cycle produces the respiratory rhythm, which is basically your respirations per minute where nothing else is affecting them. You probably meet that definition when you're sleeping, because everything in life, stress, causes faster breathing. Anyway, the medulla oblongata in your brainstem, it has those pacemaker cells, right? And these are the cells that control your diaphragm and your intercostal muscles. And they do that by way of the phrenic nerve. Your phrenic nerve is the sole innervation of your diaphragm. That's kind of scary, right? One nerve. And your phrenic nerve comes out of your cervical plexus, not your brachial plexus. Remember, plexuses were the 4 different ventral plexuses of nerves that left your spinal cord. There was the cervical, brachial, lumbar, and sacral. And your phrenic nerve comes out of the cervical plexus of your neck. The dorsal respiratory group, or the DRG, does not set the rhythm, but it can modify the rhythm by influencing the rate and the depth of respiration. The dorsal respiratory group, or the DRG, it's tasked with interpreting all the sensory inputs and organizing them. So here's some sensory inputs, right? And there's some coming up from the spine. There might be some coming down from the big brain hat. But the DRG organizes them and decides how to modify depth and rate of breathing. The DRG can receive information from ascending spinal tracts, carrying information from chemoreceptors or baroreceptors, but it also receives input from higher brain centers, like the limbic system, your emotional brain, or your cerebrum. While the VRG is setting the rhythm, the DRG is receiving all of this input. And the DRG and the PRG, the pontine respiratory group, are coordinating all of this input. The PRG is really important in receiving input from higher brain centers. It does so more than the DRG, but the pontine respiratory center is more concerned with things that you do often, like changing your respiratory rate for sleep or exercise or laughing or crying, more about like reflexes rather than voluntary. Recall that carbon dioxide is acidic. And therefore, many of our chemoreceptors that influence breathing, they're not sensing carbon dioxide or oxygen. They're actually pH sensors. This is especially important in the cerebrospinal fluid filling the central canal and the ventricles of your brain. Central chemoreceptors are the ones that are involved in monitoring your central nervous system. Peripheral chemoreceptors are strategically placed to monitor the blood in the large elastic arteries leaving the heart. The carotid bodies are such chemoreceptors, and they monitor the oxygen and carbon dioxide content of the blood going to the brain. And I have this picture here of the carotid arteries. Aortic bodies are also chemoreceptors that are placed right on the aorta in order to monitor the blood leaving the left ventricle. And both of these are concerned with the pH of blood going to the body. Both of these types of chemoreceptors feed back to the respiratory centers in the brain by way of the vagus nerve and the glossopharyngeal cranial nerves. There are other chemoreceptors that are specifically placed at the opening of the nose and the mouth in an effort to sense any irritants. Your olfactory cranial nerve 1 also feeds back to the dorsal respiratory group, which might initiate coughing if it senses an irritant. There are also baroreceptors, and they're kept in feeding information to the dorsal respiratory group. The inflation reflex is also called the Herring-Breuer reflex. It prevents you from overinflating the lungs. It's actually a reflex that controls a very special point at which your brain tells you, stop, enough inhalation, switch to exhalation. All of these inputs, the chemoreceptors, baroreceptors, emotions, they all find their way to the DRG or the PRG. And they all have their say in adjusting the rate and depth of ventilation. So there's no one breath that's the same as the breath before it. And if you're talking about a resting respiratory rate, it's almost impossible because all of these inputs are making small adjustments to the rate and depth of breathing all the time. It's kind of like how blood vessels were managed as well. At first look, the concept of ventilation, breathing, moving air, perfusion, getting blood to the area, right? Ventilation, perfusion coupling seems very confusing. It is not. It is quite simple. Every time you take a breath, there's a certain percentage of your alveoli that will not expand, right? We know that from spirometry. If the alveoli that are not going to expand, if they're going to be capable of exchanging gases in alveolar respiration, then I don't want to send blood to the capillary beds surrounding alveoli that don't inflate. We can also say this another way. Every time you take a breath, there's a certain percentage of your alveoli that will expand with that breath. Those alveoli will be able to aid in alveolar respiration by exchanging gases because I will send blood to those alveoli. Ventilation is just a word that refers to breathing. It just refers to the expansion of alveoli. Perfusion is referring to blood flow to said alveoli. This in the middle is perfect. I have an inflated alveolus that has been ventilated with air. I also have a blood vessel down here that is perfused with blood. Over here on the right, this is showing you an area where ventilation and perfusion is not coupled together. You are getting air into the alveoli, but there is no blood flow into the vessel. That's not good. You don't want to do that. You also don't want the opposite, which is over here, where no air is getting into the alveolus. It is not expanding, but you do have lots of blood running through. And look, this blood is not getting oxygenated, right? It is not turning red as this oxygenated blood over here. So this is kind of like a game of whack-a-mole. When you have vasodilation of capillary beds, ideally you also want to have bronchodilation leading to the alveoli with those capillary beds, right? If I'm going to have air, I want to have blood. If I have vasoconstriction, restricting blood flow to alveoli, then I definitely don't want to shunt air there. I would also want bronchoconstriction. So again, this comes back to the very simple statement, I want to send blood to where the air is.